I do biophysics. I mean, I look at problems in the nervous system actually, and sort of systems level problems. And my sense is that if you want to work in biology, actually, if you want to do anything in experimental science, I think the best training is in physics because it's broad, right? You learn how to do things. So I was an engineer undergrad, and the department was very nice. This was out in Illinois, and it was kind of fun. But then I was a work-study student, and I went to work at Argonne National Labs, and I worked on the particle accelerator there, making like a detector. This is quite some years ago, and it sort of became clear that it was the physicists who knew the most amount of stuff. They knew fluids, they knew electricity, they knew mechanics, they knew how to do everything. So I decided that when I went back to school, I was going to try to finish up in engineering as quickly as I could, and then I was going to shift to physics. And that seemed, okay, so that seemed great. So then there's an interesting issue. So I'm going to probably end up insulting a few of my colleagues, but you don't have to tell them this. So the easiest thing to do was to join a high energy physics group. So I already had a little bit of experience, but I was on the engineering staff at Argonne Lab. So I joined the high energy physics group because I think this is great. These are some of the most amazing machines that have ever been built. And it was absolute misery as a graduate student. I mean, it was really, I was sort of socialist streak. And, the old professors would be in the front of the room and they're walking back and forth and they're doing the thing of scratching their chin and trying to look intellectual. And then the assistant professors are sitting behind them, and then the postdocs are sitting behind them. And then the graduate students were like small children. They should be seen but not heard. We're sort of in the back of the room. And they had me gluing wires on frames, these particle detectors. This sort of first summer, I started working with them, and I thought they would like me because I figured out how to pre-stress the frames and done all these mechanics calculations, and they were still treating me like a small child. So I left high energy physics. I think it's a miserable place for a graduate student because you're like 1 out of 1,000. But the thing I found that was fun is I did a short stretch in, it was actually a biophysics lab, but it's not so much the biophysics part, it was the idea of doing tabletop physics. So I realized that some experiments, obviously, in some ways, high energy physics is the pinnacle of physics in many ways, or even more modern experiments like LIGO or large experiments, they're really at the pinnacle. But I think you have to think, the lesson from this is you got to think not just about the topic, but also the style of work. So I really like having my own bench, and in the lab today, like each student or each postdoc, sometimes they work in pairs, but usually they have their own bench and they have their own gizmo. I'll take my laptop around the lab at the end and show you what I mean. So some students build things, or postdocs build things, and in some cases I build the thing and they use it, but then they could come in every day and think about their experiment and do their own science. And it's, so that's a style. Other people like to be part of a big group. And if you look at LIGO's kind of interesting. There was a design group. There were 2 design groups, one at MIT, one at Caltech, who worked on this. I remember the day it was proposed. It was about 25 years ago. I was at Bell Labs then, and nobody quite believed it would work. People sort of trusted Werner Rainer, who was the chief designer, is a sort of a really brilliant instrument builder. So they made LIGO 1 what was incapable of detecting gravity waves, but it was sort of proof of concept that you could actually make a very, very sensitive interferometer. And then LIGO 2 obviously became somewhat famous. And the sweet thing now is that the upgrade to LIGO is using this thing called squeeze phase light. We could push the uncertainty of one phase of light into the other phase. And it was actually my boss, the guy who hired me at Bell, who did the first experiment, something like, well, it's bumping up on, he did the experiments in 83. I got there in 85. And all of a sudden now it's part of LIGO. You know, he had a little improvement, and you know, the typical thing, just apparatus just held together with the equivalent of duct tape and bubble gum on a big optical bench. But now, you know, they went from 1 dB improvement to 20 dB improvement, let's say order of magnitude, and that's the next LIGO. So it's sort of fun to do the tabletop stuff. That's a thought. So the reason I like biophysics is biology is really a frontier. I think in many ways, the logic of how genetic information is transcribed is kind of interesting. I mean, it's both storage information in a sequence. Oh, you can't even see me. Okay, I got the wrong view. You're storing information in a sequence, but then the strand of material has to fold on itself, and sort of the mechanics, the different parts of the DNA coming together are sort of part of this information transcription. So that's a puzzle, right? How physical forces come into solving information. And I went in towards neuroscience, and that's another puzzle. I mean, if you stay at home at night and drink a beer and think about it, it's a little crazy. It's matter coming together to the point that it's intelligent, and it can try to understand itself. And we're very, very, very far away from that level of understanding, but how individual elements, nerve cells, brain cells, can come together and compute is a fun puzzle, right? So it's really there's a couple of theories out there to make a little progress on. And I actually teach a course in the winter, and I try to develop, the few things that I think are on solid ground. But the whole notion of taking in data from the world, of nature having constructed sensors, building sensors, you're remembering things, using that to control a motor system and navigate in the world. And on top of that, do very abstract thought, which is why we're all here in the first place, is kind of wild. So we're working on problems in the nervous system. And so I'll give you I thought I'd give a little taste of what we're working on now, okay? But before then, I got some pictures of what different people in the group are doing, right? And I could try to explain what's going on. So let me, and the other thing is you should just interrupt me like nonstop, because there's no problem. Okay, so let me get my little thingy. All right, let me try this. I don't want to turn this into like a Skype meeting. It's too formal, but I'll, okay, so let me try to share something. Okay. Can you guys see this PDF?
Yeah, we can.
Yeah.
We see all the thumbnails. And now we see a photo.
Okay. All right, let me just get out of here a second. So let me just give a little bit of background. There's a fair number of people, there's about 7 or 8 of us doing some aspect of biophysics, from pure theory to experiments. I got a couple of my colleagues to give me some slides. Okay, this one's impossibly complicated, so let's not worry about it. I think there's a very classical part of biophysics is understanding how proteins work as little machines. So Doug Smith studied this in his laboratory. The chief tool that he uses is something called optical tweezers. It's a way of trapping particles that have a high index. This has been a very powerful tool to the study of forces between molecules. I'm going to go to the board. I'm going to just try to sketch this. Okay, so let me Let me push a button here. Nope. All right, let me push another button. Let me stop sharing, and then I'll come back. So, if you guys, can you guys hear me?
Yeah.
Yeah.
Let me just give you a little bit of an overview. So, if you decide to think about working, I would think if you want to go on in science, I'm assuming all of you do, I would encourage everyone to get involved in a laboratory in some fashion. I realize it's difficult now because of COVID, and this is not going to change, you know, for another year or two, but try to make it work somehow. So one interesting, you know, question always is what are the forces between two molecules? You have two molecules and they'll tend to bind to each other. You want to know with what strength they bind to each other. Like it could be, this could be like a receptor for a neurotransmitter and the small molecule that binds to it. So you would really, you know, you really just want to put a force on it, maybe like a spring, and just pull and pull and pull and wait till it breaks apart. So some years ago, around 1980, Art Ashkin, who was also at Bell Labs, at Homedale and Bell Labs, came up with this concept of the optical trap. And you can actually build one of these. You can take Physics 173, this lab class. And the idea is you come in, here's a lens. You turn a lens like this, where the lines cross, people think you're a professional, so you can impress people. And then light comes in, it comes to a focus. But what's going to happen is there's opposing forces. There's a little bit of radiation pressure, which is trying to push this like glass object away. On the other hand, light bends according to Snell's law, and you get a little bit of a force that's trying to pull this glass in, and it's actually trying to pull it in from top and bottom as well, and it traps it, and it's kind of fun. So with a bright enough laser and a sharp enough lens, you can trap a particle. And Doug Smith will use this to do things like okay, this is the age of the virus. He'll take like a T4 virus, and the T4 virus is trying to hack DNA into itself, and there's a protein here which is acting as a motor. And what Doug will do is he'll put a ball on the end of DNA, and he'll try to measure how much force this motor could exert, and really characterize the system. So these are very much biology questions, what the forces are between proteins, but the way that this was solved was using a tool from physics. It was using a concept of the bending of light, you know, Snell's law, ancient concept, and it was using a modern tool, not so modern anymore, like a laser, right? A tool from the 1960s, okay? And I mean, today you just buy them and you get amazingly bright, good lasers for, you know, $10. So this is a part of molecular biology that just owes itself to physics. So that's one thing. Other people do theoretical studies like this. Olga Dudko does theoretical studies with single molecules. Nina Kasselberg teaches one of the advanced biophysics courses on cell structure. We'll think about actually how many proteins come together in theory. Okay. Let me see what other pictures I have. Okay, the last picture was terribly complicated. Let me see if anybody gave me anything simple. It's the problem with academics. They try to put too much stuff on every picture. Okay, this one's kind of pretty. Let me try this one. All right, let's go wild. Can we see? Can you see Lena's picture? There's Lena.
Yeah.
Yeah. Okay. So Lena is looking at, just getting a little closer to my heart. Again, it's interaction of molecules with sort of big molecules. I mean, the mesh, the mesh inside cells, the sort of scaffolding that keeps it together. And I mean, here it's a little more interesting. You pull on a little piece of the scaffolding and the whole cell knows what's happening. I mean, how do you think of touch? You know, how do you convey forces at a distance? It's partly through scaffolding. And then I think I have one more. Okay, this one. So Alex Grossman is another member of our department. He'll probably teach 173. Alex builds instruments. We collaborate together. He's really one of the best instrument builders around. He knows a lot of fluid mechanics. So some of the, I mean, this is another aspect, you might say, where physics has had a very big impact in designing instruments. I'll get to that a little bit myself. And so in one way, you know, the classic picture is you see, oh, I'm gonna go back here. You know, you open up a textbook and you see a petri dish, and it's about 4 inches in diameter. But I mean, that's really not how bacteria live. They live in tight spaces. So Alex is able to fabricate devices that are maybe 20 microns high, just enough for a bug to slip in, and then there'll be thousands of them. So it's I think one thing that physics has added to microbiology is the idea that you can study bacteria in confined spaces. You can study bacteria, huge numbers at the same time of bacteria. I'm gonna stop this a second.
I stopped the sharing already.
Okay, yeah. So a little bit, one of the things that you can think about right from this experiment, right from Doug's experiment, is physics has allowed us to study biology at the level of one protein, right? This is sort of a new concept introduced to biology through physicists. And physicists have now, by building, you know, very, very large numbers of whatever you want to study your organisms in, allow you to really get good statistics over huge swaths of a population, which is important because, you know, psychology professors might think that things are Gaussian distributed, but nothing in this world is Gaussian distributed except for, I don't know, nothing. Okay. Air molecules in the room, okay, assuming the room doesn't leak, things like that. Right. So in my lab, we do things related to neuroscience, and let me just pose a problem here. So, I mean, everybody knows about sort of training based on reward. Does everybody know Pavlov's dog? If anybody doesn't know about Pavlov's dog? Can I go over this?
Yeah, maybe a brief summary.
Okay, all right. I'm going to show a little picture then about Pavlov's dog. All right. So let me get my sharing back. All right. This is a cartoon. Pavlov's dog experiment is this. One day I was walking around the house, I was looking for our dog, Ava. And my daughter Zoe says, just kick the food bowl and she'll come running. So that was a cue, right? The dog is rewarded with food, but the sound of the stainless steel bowl just moving was a expectation cue to the animal that there was going to be a reward. So so normally so this is just a real cartoony thingy. You give a reward, okay, which could be food or a good grade on an exam or whatever, or your parents buy you a car because you got a good grade on the exam, and that's a reward. And there's a molecule in your brain, a very famous molecule called dopamine. And dopamine all of a sudden jumps up, right? So there's all sorts of pop psychology about how to increase your dopamine concentration, and this is a good thing. But it just pops up. And what happens is if you introduce a neutral cue, like something that normally is not seen as a reward, like a tone, okay? Or kicking a stainless steel bowl. If you're a dog, but you don't know that the bowl is somehow where your owner is going to put food in the bowl, it's just a cue. But then over time, this dopamine signal moves from the time of the reward to the time of the cue. And this is a signal that's just throughout the brain. It's just and it could affect how the connections between neurons, like the little switches, sort of like the FETs between neurons, okay, how strong they are. So this was, even though that Pavlov did his experiment and he found when he gave food, the dog would salivate, and then he would give a tone and then wait a certain amount of time, 5 seconds, 15 seconds, and then give food. And then gradually he found that the dog started to salivate. I'm just gonna switch monitors here a second. Okay, give me a second here. Okay. How do I, okay, I could do it. Okay, I want to be looking at you guys, having a look at a blackboard. Okay. Are we back?
You turned your video off.
Yeah, here we go. Okay. Oops. Nope, come back. Okay, it's coming back. Okay, so why are we interested in this? On the one hand, this is something that's very special about the brain compared to the way computers work, is that things are plastic, is that you are very good at making connections between events. And just hardware, the way you wire up, you're thinking of wiring up field effect transistors, op amps, whatever, just doesn't have this sort of sense of plasticity. So this effect, the fact that what Pavlov found over 100 years ago had a biochemical basis is relatively recent. I mean, this was found in the early 1990s, right? Relatively recent. Okay, so then there's a problem though. This signal comes with a reward and then there's a cue, right? That always happens before the reward. So how does this biochemical signal come earlier? Anybody know? Anybody see the problem? It violates something, right? It seems to violate causality. How could you know that there's going to be reward coming due to this neutral cue? So people, of course, predicted that there's going to be a memory signal, and has a fancy name in biology. It's called an eligibility trace. And it was a hypothesis, came out of psychology, actually came out of computer science. Okay, and people have looked very hard for this memory trace, and it's not clear if they found it. Okay, but the one thing you know is that this dopamine signal moves from the time of the reward to the time of the stimulus. Okay, so that was a fun fact. So we had a thought, okay? And since nobody could find this memory trace, another thing that physics teaches you, which again is kind of funny that you just don't see in biology books, but this is changing now, is you're used to random events, right? You're used to diffusion. Particles are always bumping into each other. Events are, if you're away from zero temperature, there's always thermal events. Things are always bouncing around. Air molecules are crashing into each other. Dust particles are moving. And nowhere is this more evident than in biology. So, you know, the thermal scale, the thermal energy scale is about 25 millivolts, and the transition energy for proteins, the transition energy for signaling is also just a few times, you know, KT, Boltzmann thermal energy, around 25 millivolts. So it's a very, very noisy world. So we thought, well, maybe what's happening is that biology is thought of all these signaling in very deterministic way, but maybe there are some spontaneous signals, some spontaneous dopamine events that occur, and these just happen to get attached to one of these cues. And so we decided to go ahead and look for these spontaneous events, because pairing of events in time is really one of the fundamental signatures of a nervous system. So then the question is how are we going to do this? So we asked a simple question. Is a release of this molecule throughout the brain, is it always deterministic, or can we find spontaneous events? And the other thing that was driving us is a large part of the lab now for many years, you know, we look at how animals, you know, process sensory information coming, if they're, you know, these are rodents we use in the lab, they're coming into their whiskers, they're coming into their lips, they're coming into sensation through their nose. We've had a lot of fun finding out how things like breathing, just the regular breathing is a clock that drives these inputs. It's all been pure engineering in many ways, reverse engineering, but what drives an animal to be curious? What drives you to be curious, okay? Because, and the thought was, well, there's a problem in understanding how you have these memory traces, but then there's also this notion of how do you drive searching? How do you drive curiosity? And we thought maybe your own brain is playing a trick on you, and it's always increasing this chemical. It's always increasing dopamine, and that's telling you there's a reward out there, even if there isn't. So you're searching for something, right? You're going ahead and being curious, and then maybe every so often you'll actually get a reward, and that reward could somehow then be linked with this early sort of, you might say, false clue of dopamine. So crazy idea, and what the heck, right? What's tenure for? Not to do crazy ideas. Okay, so we have to make a machine to find dopamine. So did your parents show you this movie, Back to the Future? Anybody ever see that movie? No?
Yeah.
Okay, all right, I don't know, I thought it was pretty funny, so. All right, well, we didn't quite build one of these mind-reading machines. It didn't quite work. We wanted to do something with chemistry. So now let me get a little bit into some hardcore stuff. So I'm going to go back to the board and just explain a few things that come from chemistry and biology. All right, so let me see if I could push a button here, take me back. Okay, we just like that button. How about that button? Okay. All right, I'm just going to go back to the board. We're going to need to do a number of things, and some things come from chemistry, and some things come from physics, and that's what I think makes doing biological physics fun. You can pull things together. So there's a small molecule called dopamine. Let's not worry about how it looks or how it works for the moment, but we want to detect it. And what better way to make a detector than to take one of nature's own detectors and steal it, okay? So nature has these molecules, they're called G proteins, and this is what happens. They sit across the membrane of a cell. There's a whole family of these things. And a molecule called dopamine will bind to it. And then what it causes the release of something called the G protein. Okay, then a lot of stuff happens. It's interesting stuff, but let me just take that one moment. Can you see this corner of the board?
Yes, we can.
Okay, so let me just remind you about a cell. So in high school, they told you that was a cell. Okay, but that's a lie. The cell is really a shell. And this is what you call the inside of the cell, the cytoplasm. And this guy is called the endoplasmic reticulum. And so the cell is really a shell. Forget about the nucleus for the moment. And this is the outside world. And the outside has to communicate with the inside. And the way the outside communicates with the inside is through one of these G proteins. And in this particular case, we've cooked things up in a way that if dopamine binds.
Professor Kleinfeld?
Yeah?
There's some beeping in the background. I didn't know. It gets a little loud.
Oh, I see. All right. This was a phone call. Sorry. So, this was a discovery by a guy named Michael Burridge in England. And again, it's a nice story because he went out to find a molecule that contributes from the outside to the inside. People thought he was crazy because it was impossible to find, but he found it, became famous. He buys all sorts of fancy clothes now. He looks great in a Tom Ford suit, I have to say. And what happens is that this guy causes a molecule called calcium to be released. Now where do we come in? We want to find dopamine. Now, there was a fellow named Roger Chen who died rather young, who was absolutely brilliant chemist and collaborator. And what he found was the following, is that he would have a molecule, okay, just going to draw a circle, okay, and when you shine light on any molecule, you get light of a slightly longer wavelength that comes off. This is called fluorescence. And Roger devised a number of molecules that were very sensitive to binding, say, calcium, which is a fundamental molecule in biology. And it would change the intensity of the light that comes off. So you would have something that looks like this. You would have intensity as a function of the concentration of calcium, and you get a curve. So we made use of what molecular biologists taught us about these particular receptors, these G proteins, and we made a cell that had the right G protein and had one of Roger Chen's sensors, actually one of his student sensors, in this part of the cell, the cytoplasm. And now what we did is we made a little machine, a little cell machine that would do the following. When we look at the extent of fluorescence as a function of the concentration of dopamine, actually, and you get a curve that looked like this. So we had a sensor, and we could bury this sensor in the brain of an animal. So all we had to do was shine in light and then measure the light that came back, okay? And that would tell us how much dopamine the animal was producing. So that's pretty cool because now we can use microscopy and light. Right? So now there's a second trick, okay? So this is a trick that came from chemistry. Now the problem in using a microscope in the brain is that it's dense. Okay? It's like looking into your finger. The light is scattering all over the place. So you need a trick to get away from the scattering. And this is a trick actually invented, this was invented around 1980, wait, is this true? 1990, okay, by actually a guy I'm close friends with now, Winfred Denk, and we just described the technique. This comes from physics, and again, it comes from laser physics, and it comes from nonlinear optics. So soon after the laser came out, so Maria Feher, who the physics building is named after, her thesis work was on the absorption of multiple quanta by molecules. So you'll see where this is going. So we have the one thing we have a tool that can measure dopamine, and we just have to be able to measure this tool inside a rat brain. So what Maria Feher calculated was this. You have a molecule, it has a ground state, it has an excited state, like you excite a gas, like a neon sign, okay? And it emits light, it fluoresces. And normally you think of, you can excite a molecule by coming in with light that goes from the ground state to the excited state, and it'll fluoresce that down. But all you have to do is conserve energy and conserve angular momentum, and anything else is fair. So she calculated the probability of having two quanta. So instead of like one green photon, this would be like two infrared photons, say to excite a molecule that, like some dye molecule, okay? So this becomes a very nice trick to actually see deep into structure and get rid of scattered light. So let me show you what the issue is. So, let's even leave out the mouse for the moment. Here's a lens, and I have light that comes in, and it's coming to a focus, okay? Well, what this means is that every sort of level here, okay, has the same number of photons, okay? So we have the same power, same number of photons at every level. They're just very concentrated right at the middle of the beam. Okay? So the absorption goes like the intensity, okay, which here is like power per area. Okay, so what's happening is here the absorption goes like, goes like the power, which is just the intensity times the area. So what's happening here is that the light is more intense, okay, but, okay, the area is much smaller. So the amount of absorption here and the number of molecules that absorb light here are exactly the same. Okay? Because I just, okay. So suppose now I have something where the absorption goes like something strong. It goes like not like one photon, but goes like two photons. Okay. So now what's happening? This guy is going to fall off. Okay, like 1 over the area. So I'm going to get a much stronger absorption just at this focus than I'm going to get at these two planes that are out of focus. And that's sort of a key trick. So I could shine light in, and I can have then something biological, like your hand, and lots of scattering centers. The light comes in, and it scatters away. But some small fraction makes it to this center point to the lens. And it's going to excite all the fluorophores at this point, okay? And then it's going to emit fluorescence, but I know where the beam is. So I could collect fluorescence all over this animal, and I know that it came from this spot. So I could excite my cell-based sensor of dopamine and deep within the brain of an animal. All right, so let me show you what this looks like. All right, so I'm going to go back to a share. You guys with me? A little bit? A lot of it? Okay,
Yes.
so any questions? All right, so we make a fancy little molecular sensor. So we just borrow some tricks from the biologists. And, you know, people, you take a biochemistry course someday, you'll see a curve that looks like this. It's like signal strength as a function of the, it turns out it's going to be the log of the concentration. So we can measure this molecule dopamine over about an order of magnitude of intensities, okay? So let me just go on and on. So basically, now this is the kind of setup, and I'll just take my camera later and just show you something quickly. I'll show you the lab. We're going to do an experiment. So this is, everybody sees the mouse?
Yes.
Yeah, okay. So I'll take my laptop into the lab. So basically, it's a mouse that weighs 20 grams. And then there is maybe something on the order of 100 kilograms of optics above the poor little mouse, but it doesn't know about that. Okay, and we have these cells that are buried inside the animal. And then I could do this really classic experiment. I could put on a tone. This is called the cue, or the conditioning stimulus. And then time goes by and there's a reward. So initially, day one is you just give the reward, and then you get this dopamine signal that comes up sometimes after the reward. So dopamine is basically signaling I got a reward. And then I introduce this tone, which is neutral. I mean, it has no meaning to the animal, except I pair it with the reward. And I see this dopamine signal move from being here at day one to being here at day 3 to being here at day 5. And basically, at some point, the beginning of this dopamine signal comes earlier and earlier and earlier. So all we did here was repeat Pavlov's 120-year-old experiment and repeat this fellow Wolfram Schultz's 1990 discovery that dopamine is triggering the reward. But we did it in a way now that we could get a little fancier. So the thing we could do that wasn't done before, we actually measured the molecule itself as opposed to the neurons that are giving it off, okay? And we could put the animal in a closed loop setting. So first, let me just do something simple. So here's our animal again, and this is actually looking in the brain. This should be XY, not XX. Sorry, this should be 100 microns. We're looking at the small region, and we're looking at this one particular cell, which is sensitive to dopamine, and I can measure this nice response. Okay, so the new thing that we're sort of excited about is shown here. Okay, D2 sniffer is just a fancy name for our toy. And each of these little bursts, this is a naive animal. It's never seen the apparatus before. It's bolted in place. It doesn't get a reward. And it's just sitting there spitting out dopamine. It's just curious about its new environment. So by making an apparatus that uses something from biochemistry to look at a molecule and something from physics to be able to record this molecule for hours and hours and hours, we find that a signal that's been associated with reward is actually happening all the time spontaneously. It's not just deterministic. And it tracks a little bit. The animal tries to lick, even though it doesn't know that water is going to come out of the spout. It's trying to lick this thing, and it licks it, and then that's in black, and then a little later dopamine comes out. And okay, there's a lot of statistics that we could do, but the only relevant thing here is that there's a weak correlation. That means a tendency of one signal to predict the other between dopamine and, I want to get the board back here. The camera back here. There's a little bit of a weak prediction between dopamine and the animal trying to have this motor act of licking, but basically the dopamine isn't predicting anything. All right, so now we again go back to physics, and we want to see if we could actually train these animals just sitting there volitionally to increase their concentration of dopamine. So if you talk to your parents or your grandparents, you might find out that there was this thing in the 60s, 70s, where people would try to control to become calm and control their alpha rhythms just by meditating. And what we're asking this animal to do is not really meditate, but we're going to say every time you could increase your own dopamine concentration, we're going to give you a reward. So we're going to put this animal inside a feedback loop. And the idea is that if these spontaneous dopamine events are seen by the animal as something to push it forward towards a reward, then if the animal actually gets a reward, it should increase the concentration of its own dopamine events. So we made a feedback loop. So now we have this poor mouse, and we're monitoring optically what's happening in its brain. And then we're taking that signal and we're computing the concentration of neurotransmitter in its brain in real time. And we're comparing that to a threshold level. And if the animal guesses right, then we make the task harder the next time, right? So it's like a good coach, right? You run a certain rate, you make it harder the next time. And if the animal starts to fail, we start to decrease the rate. So in a sense, the mouse is turned into a function. And the threshold is the concentration of dopamine in the animal's brain with some time constant. So again, we've used ideas, not so much from physics, but from control theory in a simple way to try to learn something about the animal. So trying to give you a little bit of a flavor that even though it's a biology question, we're using all these things in physical sciences. And then it's kind of fun. So we see something like we saw before. The trace in red here is dopamine. And this is the animal licking, but nothing's coming out, and there's no relation between these signals. And then we turn on this feedback loop. And it's kind of cool because the animal starts to lick, and we increase the threshold, and it licks more, and we increase the threshold, and it licks more. And then it says, forget it, I'm going to relax for a while. And then this threshold just kind of comes down, so we get it within capture range of the animal. And then the animal, again, just keeps on kicking up its dopamine level. And this little trace on the side, on the right-hand side, is just it's a blow up. It's this gray region here. And what it's doing, you can just see these little steps. So the animal is it's controlling its own neuromodulator level. Okay, so this is new stuff. I talked about it at a meeting about a month ago. And Conrad Fu is a graduate student here. And this is his thesis work, his finding. And I'm sure we're going to have a big fight when we try to publish it, but that's something your thesis advisor never tells you about. So then we could do all kind of statistics on this kind of stuff. We could average over many animals, and we see that over the time of our experiments, the dopamine concentration really goes up. We could look at something like the number of transients. I mean, this isn't really important. The key thing is that we discovered that without a cue, there's a signal that tells you something, a good thing is going to happen, and you could just manipulate it yourself. Okay, so I'll just get to the last piece. So the last piece is kind of important because dopamine is also associated with movement. I mean, you kind of know this from all the research on Parkinson's disease, right? It's a common transmitter in the brain. And normally what happens is you move, and after you move, dopamine goes up. And that's what I wrote here. Movement, in this case, is licking, and dopamine is behind. And then we start to train the animal. And then finally, we get to this point that dopamine predicts. And then we turn the feedback off. and all of a sudden dopamine now lags. So you really need this constant training for this to be turned into a predictive signal. Now, there's another thing that people in physics are good at, and you know, I looked at for funny reasons. I recently looked into LIGO, and there were a small number of people building the machine, and there was like 1,000 faculty in the country who actually analyzed the data. And you really need to know how to do good statistics, I think, if you want to become a good scientist. And in this case, we could also ask, well, how good is the animal actually at predicting a reward? 100%, 50%? So we could just use some of the math you're going to learn. You're all going to learn, you've already taken calculus, learned calculus. You're going to learn Fourier transforms. You're going to learn filtering. And you could really learn how to analyze your data very well and make a very hard statement that this change is significant, and this change allows the animal to go from about guessing correctly 8% of the time to guessing correctly 20% of the time. So 20% is pretty good. If you could win at cards 1 out of 5 hands, I want to be your friend. You're going to make a bundle of money, okay? So that's a story that's gone on in the lab. Now, I'm going to disconnect you guys a sec, and how do I start the video? All right, I'm going to take you into the lab and show you some of the equipment, okay? Okay, can you guys see?
Yes, we can.
Okay. Anton, I'm showing off the toys. All right, this is a bunch of BNC cables, and I think you're stuck with them. So, you see where my hand is? Okay, this is the lens. The animal sits here. This is the objective. There's, right, these are modern days, so you buy a laser and it's just a, let me try to get it. There it is, it says coherent. It's, you could get a very good Porsche for the price of that laser. But just to get an idea of what's involved, so this setup actually is a little more sophisticated than the one we used. And they're going to try to, I think we better get, this is like a cooking show. You can't quite see what's going on. This is a setup that uses something from that started in astronomy called adaptive optics. So I mentioned that we're shining light through scattering material, and we could go pretty deep just based on the fact that we're using infrared light and the molecules have to absorb 2 quanta. But we also get this distortion of the wavefront of the light that goes through. And what adaptive optics does is it pre-distorts the wavefront so that when you get down to the focus point, you have a perfect wavefront for your beam. Well, it's not perfect, but it's approximately so. So we try to, hey, Panton, do you have a flashlight? Yeah. Okay. All right, you get the Boy Scout award. Okay. Can you give me a hand? I need 3 hands. That means I need one of yours. Here, hold this up. Okay, so just giving you a sense of what's going on.
I'm pointing with a flashlight to, these are mirrors that scan a beam, okay, and are basically going to scan the angle of the light that was coming into that objective I showed you, and a lens turns angles into position. And then there's a lot of lenses here just to correct for astigmatism, things you go to the eye doctor for. There's a camera back here. Let's see. It's a CCD camera. It's sort of like the old style digital camera that you use. But this is this is part of an apparatus that allows us to visualize what the wavefront looks like on the sample. And, well, Ray doesn't trust anybody. Okay. And then my very smart postdoc doesn't trust anybody not to destroy his equipment. So there's a mirror here that actually bends, a deformable mirror that bends and changes the wavefront, just like the mirror that people use in adaptive telescopes, right, to correct for distortion through the atmosphere. Okay, and then here, I think I showed you. So, there's an animal, but just an apparatus means there are stages to move the animal up, right and left. There's stages to move the lens up and down. There's a bunch of things over here, which are photodetectors. There's a bunch of optics here to separate the light we use to illuminate the animal from the fluorescent light. Okay, thanks. And no experiment is complete without a computer. All right, so, hey, you guys have questions about anything? Hello, hello? Okay, Gillian, you're supposed to start the question and answer period.
All right. So I'm guessing you use mice because they're easy to access, etc. Do you have plans to use any other animals and study any other animals' brains?
No, but let me fill that in. The one thing I didn't mention at all is genetics, and there's a few animals. Most animals have been sequenced now. Their genome. The key is to also experimentally is to use this knowledge to your advantage. So what that might, for some people, that means like blocking a certain signaling pathway, removing a protein, so altering the genome. It also means that different cells, not just neurons versus liver cells versus pancreatic cells, but even different classes of neurons produce different proteins. And so you could use this knowledge to make only those cells produce a color, like a fluorescent molecule. So you could target specific cells, or you could have specific cells produce a molecule that gives you some functional feedback. Like I talked about calcium going up. There's just the beginnings of molecules that will report voltage. It could be other molecules that tell you about the function. And then starting, it's almost 20 years ago now, there are molecules you could put in the cell and then use light to change the property of those cells, cause those cells to fire a pulse or not fire a pulse. So the engineering principles of perturbation, where you go in and you perturb the sample, or you cut a feedback loop, are things you can now access, but you want to do this in a hand-over-hand way. Like you'll say, I think these cells are responsible for being part of the feedback loop, so I want to manipulate them, and then you'll do histology to find out if you got the right cells. So mice have extremely well-developed genetics. These things called zebrafish have well-developed genetics, not quite as rich a behavior. There's some other worms that have good genetics, not as good behavior. Flies actually have very interesting behavior and extraordinarily good genetics. And probably the wiring diagram of the fly, of the neurons, is something that's likely at least to have a first pass solution in the next 5 years. So people move towards mice because of the genetics, but if you're interested in biomedicine, that's not necessarily the best prep, right? I mean, the joke is that we know how to cure cancer in mice, but it doesn't help, right? Other questions? Yeah.
Question, Professor. So from the feedback that we saw earlier, so we saw that the dopamine release was before the actual reward happened, right? So
wait, I'm having, hold on a second.
sir, sure.
I'm going to put some earphones on because sometimes the sound isn't so great.
Yeah, no worries.
Also, my ears are totally stuffed up with water. Yeah, go ahead.
Sure. So from the feedback that we mentioned earlier, we saw that dopamine was released earlier, right? Before the actual reward happening.
Yeah, it's an expectation of reward. Yeah.
Right. But does that mean like the reward becomes less rewarding in a sense?
Okay, that's an interesting question. So, how would you determine that?
I'll say, it's also a good question. But say if consuming the reward, say, give you some dopamine rush, then if the expectation of it causes it to be released earlier, do we still release this like the same amount as we usually do by consuming the reward, or does it like diminish because of the expectation beforehand?
Yeah, that's a good point. So the experiments to date don't actually show, like you might think that the concentration just goes down and down because you're getting used to it, right? Like, I mean, but there's not that many experiments. These are not my experiments. These are by a guy named Paul Gunchmer. I mean, the one thing that the early dopamine signal does is its amplitude is very good in reporting the probability that a reward will happen. Like instead of giving the reward every time, you could give it, 5%, well not 10%, 20%, 30%, and you get a different height. But there's no, I don't think the experiment's been done properly yet, but no one's reported like an extinction that the expectation signal diminishes. And okay, the one thing that will cause it to diminish though is when the animal is sated. So I mean, the little mice can only drink so much chocolate milk or so much sugar water. I guess you and I can only drink so much Coca-Cola, and so it will diminish. But I don't, it might diminish before you're sated, right? It might diminish before you're sated. I don't think that's been answered yet. It's a good question.
Thank you.
It's a good question. Yeah.
Of course.
Yeah, please.
Did you offer the mouse the same reward every time? And if so, how did you pick what reward to offer it?
Okay, that's a fair question. So in this experiment, we did offer it the same time. So at some level, this experiment was extremely crude. It just, it was the first time through. It was actually a very talented graduate student just to get everything together. So we did a previous experiment. So in the 90s, using monkeys, the fellow who found that the dopamine signal moves from the time of the reward to the time of the cue, Wolfram Schultz, did everything in monkeys. And we previously published basically a repeat of his experiment, but for mice. So we worked out, I wouldn't say we were exhaustive, but we worked out a set of parameters that made this, the fancy word is reinforcement learning, work in mice. And, this is basically something like 100 microliters, sounds miserly, but they're little animals, 100 microliters of sugar water. And the mice would easily go on for about 100, 150 trials. And it seems stable. So I think that the deeper question is like, when you're setting up an experiment, do you know when to stop noodling? And particularly in biology, where there's just too many parameters to noodle. So I think something works, and you want to then, if you think something's a critical parameter, you want to change it, and you want to make sure that your effect is not sensitive to that parameter. So I wouldn't bet money that the experiment would probably work equally well with 80 or 120 microns, microliters. But 1 ml, I mean, forget it. I mean, the animal's going to drink 5 or 6 times and then go on strike. So, yeah.
Thank you.
Yeah. Anything else, you guys?
I have another question.
Yeah.
So since you've measured dopamine and gotten the data from that, do you have plans on measuring other things besides dopamine in the brain?
Yeah. Okay. So I'm giving you a very sterilized version of what happened. So this poor. Okay. So we, like, half the lab studies things related to the control of blood flow in the brain, and then half the lab was studying things related to sensory motor control. They actually, the circuitry for both of these, the initial circuitry for both of these actually is in part of the brain called the brainstem. So there's more overlap than it seems. So we expected that this molecule, which is structurally very similar to dopamine, called noradrenaline, was going to be responsible for decreasing the flow of blood in the brain, right? It had been messy reports. And we made these sensors initially for that reason, and we made one for noradrenaline. And Conrad started doing these experiments, and it was a mess. I mean, we knew the sensor worked, and we could put wires in the part of the brain that was responsible for release of noradrenaline, and you know, we got the positive controls were working, and but the animal would sit there, and we'd make the animal do a task, or we'd stimulate the animal in a way that we knew blood vessels were increasing in size and blood flow was increasing, and noradrenaline was absolutely all over the map. It was just, and you know, time is ticking by, and he has to graduate. And if he doesn't graduate, it makes me look really bad. So, I mean, it was just a bad deal all the way around. So then we just had this idea, and it wasn't really about dopamine. It was just asking the question, could animals change their own modulator concentration, right? It wasn't really thinking about learning and reward. So we started with noradrenaline, and it worked. It was dirty, but it worked. Not as well as dopamine. And then we said, okay, let's do what you said. Let's try other neuromodulators. So acetylcholine is something that has to do with attention. Hopefully acetylcholine is high in everybody's brain listening in. And we expected that not to work, and sort of negative experiments are dangerous, but and then we tried dopamine almost as a control, and it just worked even better. And we were starting to finish up, and it was like it was scientifically, it was the one good thing that came out of the campus shutting down in the spring due to COVID. I have a close friend in biology. He was also a physicist by training, Jing Wang, right? You might take one of his courses one day, systems biology. I would recommend him. I'm close with him. I'm close with his thesis advisor. They're really intellectuals. And he was all over me that I didn't appreciate the dopamine story, and he gave me some theory paper to read, and that maybe there's something here about reward. So I read the theory paper written by, I don't know, somebody, right? And some psychologist at Harvard. And it was mathematical, and it was absolute pure crap and dribble. And we just started arguing, and then we realized that we could take Conrad's data, and there was actually this story. So we did a few more experiments. Conrad did the experiments, and the story popped out. You know, so I'm presenting it as though in a very linear fashion, as though, but in fact, I mean, we had these ideas in the back of our head about curiosity, but I wasn't really thinking about random events that way until I got into this sort of
friendly argument with a friend of mine. So, and I think that's, you know, I think that's science at its best. I have to say, is you, yeah, you know, when my daughter was in 5th grade, they asked her, she took a science class, and she had a report on the scientific method. So of course she asked her parents, who are both scientists, you know, how does it work? And we said, well, you sort of have an idea, and you go out there and you acquire data, and you know, you find an interesting topic that you don't know much about and seems interesting. You acquire data, then gradually you try to come up with some hypothesis, and then you test it, and you take more data, and then you find out that your original ideas were all wrong, and you write it up. And she got a C-, and she ended up as an English major after this. But this is how it works, right? You know, you find an area that's interesting. I mean, sometimes, you know, in particle physics, sometimes you predict a certain particle. But I think you even scratch a particle physicist, and you find out that they think that every time you go up an order of magnitude in energy, you'll find something new. And our feeling is that if you know, in this case, certain molecules are important, and they just haven't been measured before, and you know, pick an experiment which you think should work, and if it doesn't work, there's probably something interesting, and if it does work, then you have something to say. So so yes, we measured other molecules. We also talk about noradrenaline, I think, when we write this up, but I really think the important thing is dopamine, and what we want to do next actually is have animals forage, right? Is just search for food, and then we want to look at the dopamine levels as they search, and see if that motivates them to search further and further, and then we could go in, and we know the part of the brain that's sending fibers into where we measured in motor cortex, and I could affect those neurons. So now I want to perturb the spontaneous rate of dopamine and see if that affects how well the animal forages. So that'll be kind of fun, right?
Yeah,
So
that sounds cool.
Yeah. So
Thanks.
that's sort of a modern thing, right? And again, that'll use a little bit of physics. We got to put fiber optics in. We got to do all that jazz, right?
Are there any other questions?
I have a quick question.
Yeah.
I was wondering what the process for getting the mice prepped for the experiment actually is, because I assume you have to get a viewing window in somehow, and have a, like, I don't know, some way of stabilizing the mouse so it's still enough for the setup.
Yeah. Let me just grab I'm just going to grab a paper that I know has some nice illustrations. And this was actually, there was a graduate student at UC. He was an undergrad and a grad student, and now he's a teaching professor here, Phil Tsai. Some of you might bump into him in your career. And he did his thesis in my lab, and then he stayed on. I think he thought he was going to stay for 1 or 2 years to finish up a project. It took 8 years, but you know, that's how it goes. And then he put together some of the lab classes, some of the advanced labs, but just give me a second here. And I just want to get this paper so I can show you what some of these preps look like. Okay. Okay, so I'm going to try to share this again. Okay, so as you guess, some experiments are done with an animal just walking around. It might be an animal that's walking around with wires or fiber optics in its brain, wires to record nerve impulses, fiber optics to record, like, maybe one of these molecules I talked about to measure calcium or some other signal. And then there'll be some electronics on the head of the animal. Okay, speaking again about physicists in neurobiology. So Tim Harris, who was a colleague of mine back at Bell about 3 years ago, published on the got together with this, IMAC, which is this sort of experimental lab for integrated circuits in Leuven in Belgium, and they built the highest density array using sort of standard silicon technology, and that's what people do for electrical things. So this is what we do for looking at the animal. So to make it turns out that if you take off all of the bone to make a craniotomy, so you could image through it, that this has a number of deleterious effects on the animal. So this student, Pat Drew, and as I mentioned, Phil Tsai, came up with a nice prep. So we thinned the skull down to about 20 microns thickness. You could do this mechanically, or you could do this with laser light, amplified laser light. And so what you end up with is, this is supposed to be cortex. It looks a little bit like SpongeBob. This is the thin skull. And then there's glass here. And what we found is that if we put a thin layer of cement cyanoacrylate and then put glass over this, just keeps all the oxygen out, and it keeps the skull from regrowing. And this gave us a very, very good window. And then you really want to ask how well you could do. So just to get some idea, this is, so some of you may have heard, or you will hear about this thing called the transfer function or Green's function. It's really telling you how the system responds to an impulse, like a pulse, a delta function in time, and a delta function at a point in space. So in optics, what happens is, you're basically, you don't see arbitrarily small objects. You only see objects that are down around, one wavelength. And more so in optics, what's happening is light is coming in an asymmetric fashion. Right? It's not coming in equally from all directions in a microscope. So you tend to see worse along the optical axis than lateral to the axis. So this is called the point spread function. So it was just a little tiny little dot here, but in fact, it sort of looks like a zucchini, okay? And this is a zucchini. So this is an image, just into water, perfect microscope. This is as beautiful as it ever gets, but our little system here, we put a glass window over it, it distorts it a little bit, and then we make our fancy schmancy window for seeing into the animal, and it's distorting it even more. You can see the light is really spread out. It's really not in the center of the zucchini. Sometimes it even bends a little bit like a zucchini bends. And this is, nonetheless, zucchini or no zucchini, this is enough to see some of these very fine features on a cell. So a neuron has a synaptic input. This is called a spine. This I think this thing is 5 microns. So this is about 2 microns apart. So and this is consistent with having about a 1 micron resolution. So I think that's, yeah. So the animal has this window on it, and the animal has a bar across its head, and it's bolted to a table, and the animal is free to run. And it's, in these experiments, at least after the first day, it's free to get all the water it wants. So this is called the head fixed preparation. So obviously the vestibular system is out of the picture, but for many, many things, this allows you to recapitulate behaviors you get in a freely roaming animal. And there's really, you can make miniature microscopes, people have, and you'll get maybe 10 microns resolution, and that's very good for some things. But for these experiments, they're just not adequate. So you really have to have a head fixed animal on one of those microscopes like I showed you earlier. Yeah, long answer.
Great, thanks.
Any other questions, guys?
Okay, so if you guys are, I just showed you like a little, this is just something that's exciting to us now. If you're looking for a lab to work in your senior year, junior year, or even in particular if you're thinking of going on to grad school, you should, whatever fields you think you want to go into, what I would urge you to do is go to a few faculty members that seem like they would be appropriate and download some of their papers, right? Their publications that have interesting titles. Don't worry if you don't understand half the stuff. I'm not even sure that most advisors understand half the stuff in their papers. But then just say, is this something that I would be proud of? Is this the kind of science that I want to do? Just get a sense. Are they thorough? Is it clear? Is the writing clear? Is the experiment interesting? Is it well controlled? I mean, these are hard things to answer. And do you think you'd be happy having a paper like this as your thesis project? Right. That should be the kind of thing. And that's my best advice. Whatever. But if you guys have more questions, just I'm here. I'm not going anywhere. Like I say, I travel 5.5 miles to go from home to here. And yeah. So just send me an email and I'll gladly chat with you. Okay. And I really urge, I mean, I realize the next year or so is going to be a little difficult to work in the labs. It's even difficult to work in your own lab, but you should try to get involved somehow in research, right? It could be a lab project, could be a computational project. You just, something which is interesting and something you could, that's going to force you to learn a lot of stuff that you won't normally learn in classes, and something where actually you could fail at for a long time until you get it. I mean, that's the only way to learn, right? If things keep working, you're not pushing yourself hard enough, right? It's like sports. I mean, if it's just working, there's something wrong. You gotta push yourself, right? Yeah. Okay.
Thank you so much, Professor Kleinfeld.